setting out in little chunks, little pieces, it will be a lot easier, a lot less daunting. I want to go back to this section. It was really on a little bit of the last video, but I wanted to work a couple of genetics problems with you. So if we look at this first one, monohybrid cross, black fur is dominant, gray fur is recessive. Really what I always encourage you to do, first step, is assign the alleles. So we've got the allele for black, and we've got the allele for gray. But it tells us that black is dominant, so we write that with a capital letter. Remember, an allele gets one letter. So black will get a capital B, gray will get a little b, not a G, because we're trying to represent that gray is actually recessive to black, not co-dominant. And so we're crossing a purebred or homozygous black dog. So that dog is going to be big B, with a hybrid black dog, heterozygous, big B, little b. Now we can put that in our Punnett square. Remember that when we write a parent's genotypes, this black dog's genotype, these two letters, we actually separate the two letters because this parent is either going to give the big B or this big B, but only gives one. Remember meiosis, which will separate the alleles so that each parent just gives half. So here we have a parent who has two big Bs, but they're going to only give one to their offspring. The other parent will also only give one. When we look, what is the chance that this sperm and this egg come together? This sperm and this egg, this sperm and this egg, this sperm and this egg. So there's our Punnett square, but now we have to answer the questions. The genotype ratio will actually compare the number of big B, big Bs, to the number of big B, little b's, to the number of little b, little b's. Well, we had two big B, big B's, two heterozygotes, and no homozygous recessive. And so we use those genotypes to hold the place. The phenotype ratio, we're going to compare the phenotypes. How many are black? That's one phenotype. To how many are gray? And that's another phenotype. And so black, this represents a black dog, black dog, black dog, black dog. All four represent a black dog, no gray. So the probability of the offspring being a black dog is 100%. And so that's just a reminder of how to do a genetics problem. When you work on co and incomplete, remember co is going to use two capital letters, and incomplete uses a capital and one with the R. When you do sex-linked, you do want to remember that sex-linked traits are on the X chromosome. Sex-linked traits are carried on the X chromosome. It doesn't ask you to do a problem here, but I really encourage you to go back. This was in section 3.3, so you want to go back and really look at section 3.3. I do want to work one of the blood type problems with you. I want to help you remember the genotypes. So what we should actually do is start with the alleles. There are 3 alleles for blood type, meaning if we look at the genes for blood type, those genes come in 3 forms. One form is the I capital A. The other form of the gene is I capital B, but the third form is little I, meaning this is recessive. These two are co-dominant. So if I am blood type A, my genotypes could be IA, IA. That might be hard to see, but I'll try to make sure I'm talking slowly. Blood type A, your genotype could be homozygous, IA, IA. Or it could be heterozygous. I'm going to write it out here, IA, little I. So genotype's always 2 letters, but what we're looking at here is this idea that your genotype could be homozygous for A or heterozygous with this little I being recessive. B is the same idea. You could be IB, IB, or IB, little I. For AB, you really just have one choice, IA, IB. And for O, again, just one choice, little I, little I. So then it asks you to show the possible Punnett squares for parents who have type A and type O blood. They could be, remember, if you're type A, you could be heterozygous or you could be homozygous. So you have to set up two Punnett squares. At the bottom of this page, the genetic disorders, you're just matching up some of our genetic disorders. If we move on to EOC 7, this is where we hit pedigrees. And this is a great one to review. Remember, the first question that you ask yourself is it recessive? Is it hidden? Can it jump? Can it skip generations? Here we see two parents that don't have it and a child that does. This must mean that the parent is hiding. Both of these parents are hiding. So if it skips a generation, it is recessive. So that means that this male is little a, little a. They do actually tell you that it's Tay-sex, which lets you know that it is not sex-linked. Remember with karyotypes, this is a picture of the chromosomes. So karyotype is a picture of the chromosomes. Remember, I'm going to give you extra credit if you're watching the video and writing these types of things in. When we look at chromosomes, chromosomes should be normal, means that you're in pairs, that you have two. If you are XX, now be careful, these chromosomes are single chromosomes, but XX is the name for being female. And so we look all the way down here at the bottom. We don't see two of the same size. So remember, this is XX, which is female. If I have one tall one and one little short one, that's XY, and that's male. And so when you look here, you're trying to find the genetic disorder and the sex or gender. A little further down this page is DNA fingerprinting, which is where you're going to look at the sequences of DNA. One thing to remember is that the child's DNA came from mom and dad. So let's look at this band right here, or let's look at this first one. This child's DNA had to come from the mom and the dad. This first band came from the mom, so I know where that band came from. This second band did not come from the mom, which means it has to be coming from the dad. This guy doesn't have that band. This man does. So I know that this actually has to be the dad, that male A can't be the dad. Now I do need to keep checking because what if this one doesn't match up? But this band is also from mom, so we would say that male B is the father here. Recombinant DNA, this is also going to be called a transgenic organism. And then you're going to define what that means. These are the steps of making recombinant DNA, so you just need to go back to your notes. Next we have page EOC 8. EOC 8 looks at asexual versus sexual reproduction, and so you're going back to try to remember, how do organisms make more of themselves? Asexual is where you're actually going to be making identical offspring. Sexual is where you're going to be making varied or different offspring. And sexual is because you have two parents giving DNA, and that DNA could join up in a number of ways. Mitosis, you want to go back and identify what mitosis is. Keep in mind, somatic cells are the same thing as body cells. So if you have a cut or a blister, mitosis is going to happen there to heal it. And then you do want to remember that daughter cells are genetically identical. Sometimes we call them clones because they are exactly the same as what the parent was. So if the parent has 10 chromosomes, then the offspring will have 10. I do want you to add in one extra picture here next to this, because 10 divided by 2, how can that possibly be 10? 10 divided by 2 we would think would be 5 and 5. But remember, we actually start with 10 single, and then the first thing we do is copy the DNA, and now we have 10 doubled chromosomes. So now when I divide the 10 doubles, I go back to being 10 singles. And that's how mitosis gives us 2 identical cells. And so you can see that picture here. It does say DNA replication, but I wanted to give you another diagram to add as well. The bottom of this page looks at meiosis and genetic recombination. And so you do want to be able to look at a cell and predict what will happen after meiosis. Keep in mind, if this cell has 4 chromosomes, after meiosis, we could have lots of cells, but that one cell should only ever have 2 chromosomes. If I tried to give this cell a big T and a little t, this sperm or egg now doesn't have any of the S information. So this will not work. I need a cell that has both T and S information. So we're going to stop there because that's the end of these EOC pages that you're asked to do through the weekend. If you want to work ahead, there'll be another video next week to help with the rest.